In this work, we will demonstrate a Facile protocol to produce homogeneously acetylated proteins at specific sites by using E. coli as the expression host. The reversible lysine acetylation of proteins is one of the most abundant post-translational modifications in nature. It plays important roles in a wide range of biological processes, including gene transcription, stress response, cellular differentiation, and metabolism. Generating purely acetylated proteins at design positions is very useful for studying protein acetylation. However, it is difficult for most classic methods. This challenge has been solved by the genetic code expansion technique. It utilizes an engineered pyrolysyl tRNA synthetase variant to charge tRNA pile with acetylysine, then utilizes the host translational machinery to suppress the UAG stop codon in the mRNA, and directs the incorporation of acetylysine in the design position of the target protein. Firstly, the amber stop codon needs to be introduced into the corresponding position in the target gene by site-directed mutagenesis. Amplify the template plasmid containing the gene of the wild-type proteins and insert the stop codon mutation from the primers by the PCR reaction. After PCR, the amplified material is added directly to the kinase ligase DPN1 enzyme mix for 1 hour at room temperature for circularization and template removal. Add 5 microliters of the mix to the tube of 25 microliters thawed competent E. coli cells. Carefully flick the